Hello all, and a very warm welcome to today's ISNTD Connect in partnership with the Global Schistosomiasis Alliance. It's a pleasure to welcome you all today in this month of April, a month that's very much been centered around One Health, and for NTDs, which are inherently One Health diseases, it seemed especially timely after this really big global moment earlier in the month in France, pushing milestones and really trying to operationalize what has been said at length around One Health and infectious diseases. It seemed like a really good time to take a closer look at some of the tools and approaches that leverage environmental or animal dimensions to drive the progress in the control of these diseases. And so it's my absolute pleasure to welcome today our speakers and this panel, bringing to us some fascinating work from environmental DNA and what using these new technologies and tools might mean for the control and the understanding of schistosomiasis. So as recently as 2025, the Royal Society itself reported that rapid progress in eDNA or environmental DNA research over the last decade is really transforming monitoring, disease detection, and it's moving from being quite a niche technology into mainstream science. On the one hand, eDNA really gaining traction within infectious diseases, but also combining that with schistosomiasis is a really promising approach, a disease that, as we know, has some difficulties in its surveillance, can be really persistent, is a highly focal disease, and on top of it, it really persists in quite difficult endemic settings. So it's my pleasure today to welcome our speakers. We're going to hear some wonderful science and some really interesting, very practical insights from Brazil and the Philippines, and to get a better understanding of how this scientific tool can not just elucidate some of the gaps and the question marks around schisto and schistoprevalence, but also how can this become a bit more mainstreamed into a policy aspect and policy uptake? So a very warm welcome to our speakers, very warm welcome to our audience as well. And today we will be hearing from our three speakers on screen. First, it's my pleasure to welcome you, Sandra Grossi Gava. Welcome and thank you for being here. You are a researcher in public health at Fiocruz, Minas, which is a regional unit of the Oswaldo Cruz Foundation in Brazil. Welcome. We will then, and we will hear from you a really nice introduction around eDNA. What is it and how does this apply very nicely to schisto detection and control? We will then welcome and hear from you, Joseph Valencia. Welcome, Joseph. Nice to see you.
Yeah, nice to see you. And yeah, thank you for our participants.
Thank you so much. It's quite late your end. We're really grateful that you were able to join us. You are representing the Graduate School of Health Sciences at the Niigata University in Japan. You're also with the Research Institute for Tropical Medicine at Metro Manila in the Philippines. Thank you for being here. And Mark Revolteado, Mark, you're going to talk to us a little bit more about water-based environmental DNA. And you're joining us today from the Graduate School of Medicine, Dentistry, and Health Sciences, also at Niigata University in Japan, bringing us a lot of insights and learnings from your work in the Philippines. So thank you to the three of you for being here. Big thank you to everyone who's made time to tune in. An apology because my voice is quite hoarse today, so I will now bring this intro to a close and hand over to our wonderful speakers. Thank you for joining us today. And Sandra, the floor is yours.
Thank you, Mariani. Can you see my screen?
Perfect.
So good morning, everyone. Greetings from Brazil. First, I would like to thank you, the GSA team, especially David, Anouk, and also Martina, for inviting our research group to join this presentation. I also thank Marianne for organizing the session together with the ISNTD. And today I will share some of our research results on environmental DNA for schistosomiasis detection and control, showing some advances from Brazil. As Marianne kindly introduced me, my name is Sandra Gava. I'm a researcher at Fiocruz, Minas, and I'm working in the immunology and medical molecology group that's led by Dr. Roberta Caldeira and Dr. Marina Mourão. Just to give you some context, schistosomiasis is a neglected tropical disease endemic in 78 countries, mainly in Africa and South America. And the preventive chemotherapy with praziquantel is still needed for about 253 million people in 50 countries. Here in Brazil, we still have about 25,000 cases per year, mainly in the states of the Northeast and in Minas Gerais. And we had the presence of 3 host invertebrate species that serves as intermediate hosts of the parasite. To achieve the elimination of schistosomiasis as a public health problem, we rely on 4 main pillars. First one, the environmental sanitation, with access to sewage systems and treated water. The diagnostics, with the detection methods with high sensitivity and specificity. The treatment that currently is only one drug available, that's praziquantel. So there is a need to develop new drugs. And fourth, the control of the intermediate host that includes the search for new molluscides, environmental interventions, and biological control strategies. In 2022, the WHO published a guideline on control and elimination of human schistosomiasis, and this guideline was reviewed by the Fiocruz group. That is a group of researchers from Fiocruz, Minas that works on schistosomiasis, and I'm sharing the paper with you. From these recommendations, I would like to highlight that in the Brazilian context, there is a need for investment in research, especially for testing in humans and in snails, and to monitoring the presence of Schistosoma mansoni to confirm the interruption of transmission. Our research group also hosts the National Reference Service for Schistosomiasis, where we receive snails from all over the country, and these snails are examined for circadian release after light exposure, and the negative snails are also crushed between plates to detect the parasite in the prepatent stages. In addition, we also do a molecular method based on LS-PCR for schistosoma detection. However, these current methods have limited efficiency and accuracy. There is a very difficulty in snails collection and transportation to the lab. It's difficult also to detect the parasite at the prepatent stage, and these techniques are very time-consuming and require specialized and well-trained personnel. Here in Brazil, we had a lot of areas with low endemicity and low parasite burden, which makes the detection of this transmission force even more challenging. So eDNA emerges as a tool to circumvent these limitations. So what is eDNA? So environmental DNA, or eDNA, is the DNA released by any organisms into the environment. It's sensitive, non-invasive, and is applicable to ecology, conservation, and public health. Sources of eDNA includes skin cells, feces, mucus, and urine, or any other source of genetic material. And it can be collected from the water, the soil, or even the air. So the key idea is very simple. We can detect organisms without seeing or capturing them. And how does eDNA works in practice? So from a collected environmental sample that can be water or soil, we first extract the total DNA from that sample. Then we use some amplification and detection molecular methods to amplify the target of the organism we are looking for. So the interpretation is also very simple. If DNA is detected, it means the organism is present or was present very recently. Here I'm showing some examples of studies using eDNA for schistosomiasis detection. These works were mainly conducted in Africa, so this approach is not new. However, until recently, there were no studies from Brazil using this approach. This is important because transmission settings can be very different between Africa and Brazil, so it's necessary to validate these tools in the Brazilian context. To do this, we used primers described by Sato and colleagues that targets the COI gene from Schistosoma monzoni, and we evaluated the specificity of the detection using DNA from Cercaria, from other trematodes that occurs in Brazil, and also the DNA from other Schistosoma species, and a lot of samples from diverse snail species that we had at our medical malacology collection, performed qPCR, and analyzed the results. So by qPCR, we detected DNA only from Schistosoma monzoni, Schistosoma hematobium, and Schistosoma mathiei samples. It's important to note that only Schistosoma monzoni occurs in Brazil, so this level of specificity was appropriated for our context. And we did not detect any amplification from DNA from any of the snail species, human DNA, or the trematodes tested, which shows the good specificity of the assay. Next, we standardized the filtration and DNA extraction steps. We used different types of water samples, like filtered, dechlorinated, water from tanks containing only Biomphallaria glabrata, and also water from tanks containing Biomphallaria glabrata that was challenged with Schistosoma monzoni. Then we did the filtration, DNA extraction, and qPCR. Here, we decided to include two new molecular assays to analyze the samples. So we included the LS-PCR we used at the National Reference Service and also a lump assay. Here you can see the results from LS-PCR, lump, and qPCR. As expected, we detected Schistosoma monzoni DNA only from water containing Biomphallaria infected with Schistosoma monzoni, which confirms that the DNA can successfully detect the parasite in these controlled conditions. Next, we move to field validation. We are conducting studies in two endemic states in Brazil, Minas Gerais and Alagoas. At each site, we performed water filtration and transported the membranes to our lab. In the lab, we do the eDNA extraction and the molecular assays to detect the parasite. It's important to note that we are also conducting snail surveys at the same sites at the same time, and we started to measuring some abiotic data like air temperature, water temperature, dissolved oxygen, hardness, and conductivity of the water. Here you can see our first study area. The map shows the 9-point sampling points in the Jiru district of Joaíma, here in Minas Gerais. And I'm also showing some photos from the field work to give you an idea of the environmental conditions. And looking at the results, with LES-PCR, we did not detect Schistosoma monzoni DNA from these field samples. So as LES-PCR was not suitable with these field samples, we decided not to use this method in the filter sites. With LAMP visualized by CyberRing, we detected Schistosoma monzoni DNA at sites 3 and 5, and using LAMP visualized by geoelectrophoresis and using qPCR, we detected an additional site, that's site 4. Important to note that the malacological survey shows Biophalaria glabrata shedding Schistosoma monzoni cercari at sites 3 and 6. Another study area was located in Ribeirão das Neves, also in Minas Gerais. You can see the 3 sampling points on the map, as well as some photos from field work. In this area, using LAMP and qPCR, we did not detect Schistosoma monzoni DNA at any sampling site. It's important to note that the malacological survey had no positive results either. So just to present you a summary of our findings and a comparison with the malacological survey. We observed an agreement between the eDNA method and the malacological survey at 2 sampling points, and one additional site was detected only by qPCR and LAMP. Here you can see the results that were published last year, and a QR code for access. Our main finding is that eDNA can detect the Schistosoma monzoni DNA, even when traditional methods do not. However, we cannot tell if this DNA comes from Miracidia or Cercari. So this raises an important question. Where is this contamination coming from? To better study this, we are doing also microbial source tracking with the samples. So we are using microbial source tracking for tracking sources of fecal contamination using qPCR. This approach detects specific markers, microbial markers from host specifics. And this may be important because these hosts may play a role in the parasite life cycle. We are using the same samples we collected for eDNA to obtain more information from each of the sites, and we are performing qPCR to detect universal, human, and muskrat bacteroids, and we are also testing for Ratus and Ratus norvegicus directly from the DNA, and we are currently searching the literature for new assays to investigate other potential hosts. Just to give you some idea of our ongoing work, another study area is in Alagoas as part of a larger project called Saneia Xisto. Until now, we have sampled 77 water bodies, 18 was already analyzed, and we are conducting also environmental data collection, like abiotic data and the snail survey. And in this project, we are also collecting host data through the examination by cat-to-cats of a lot of people and conducting questionnaires on sanitation conditions at both household and community levels. So this work is ongoing. Here you can see the locations where eDNA was and snail surveys were conducted, and at this moment, the eDNA samples are being processed. Just to give you again a brief summary of our eDNA projects. So far, we have analyzed 37 sites in 4 locations in Minas Gerais and 10 locations in Alagoas. Among the samples, we have 10 positive for Schistosoma monzoni, 18 positive for human bacteroids, and the statistics show no significant association between these 2 variables. The malacological survey showed the presence of the invertebrate host at 24 sites, 5 of them shedding Schistosoma monsonis cercaria. The statistics show a moderate agreement between the eDNA methods and the malacology method, with good overall performance of eDNA and a strong ability to rule out the negative sites. It's important to note that this data is still being analyzed, so these results are very preliminary. As I mentioned, for some sites, we also collected the environmental data, including air temperature, water temperature, pH, dissolved oxygen, hardness, and conductivity. This is still ongoing working, so these environmental data are currently being analyzed, and we aim at identify correlations with the presence of Schistosoma monzoni and the host's presence, and maybe to explore some predictive models. To conclude, we successfully standardized eDNA in our lab, including sampling, filtration, DNA extraction, and qPCR, LS-PCR, and LAMP assays. Our results shows that eDNA is a sensitive and useful tool that was applied in the field settings. And for the next steps, we will also expand the microbial source tracking to better understand the role of non-human mammalian hosts in maintaining transmission. And finally, we aim to develop or identify some specific assays to detect the biofilaria DNA directly from the eDNA samples. So together, this approach can improve schistosomia surveillance and support the elimination. And finally, I would like to thank you, our research group, especially the team involved in the eDNA projects, Dr. Roberta Caldeira, Marina Mourão, Lanja Montresor, and as well as my 2 undergrad students, Isadora and Bento, that do a lot of lab work on these projects. I also thank our collaborators from Fiocruz and also from different institutions, our funding agencies, and all of you for your attention. Thank you very much, and I'm happy to answer any questions at the end of the session.
Thank you so much, Sandra. Congratulations to you and the entire team behind this absolutely fascinating work. You're getting lots of very positive, interested feedback in the chat. Halata al-Bashir is saying that was great work and lots of comments. Thank you so much for really explaining this shift from quite a labor-intensive snail surveys to showing us the scalability of it and what's needed in order to boost innovation and practical innovation in this field. It's been wonderful. And just before coming on live, Joseph, you did ask whether there would be many questions, and my answer was yes. So they are coming in fast and thick, and I think we'll stick to the plan, which was to take all the questions at the end. But please, if you do have questions, either pop them in the chat or in the Q&A function, and we will get to them as soon as possible. But in the meantime, moving from Brazil to the Philippines, and looking a little bit more closely at the mechanics behind water-based eDNA approaches and soil-based eDNA detection as well. And maybe that will start to answer a little bit more of those technical questions that are coming through. It's my pleasure to hand over to you, Joseph and Mark. Joseph, you first to share with us some of your work on soil-based eDNA detection from the Philippines.
Okay, let me share my screen. Okay, so good evening from Japan. Good morning from in the other side of the earth. So yeah, so Thank you for coming for this webinar. So I'll be presenting our study on soil-based environmental DNA detection of Oncomelania hupensis quadrasi and Schistosoma japonicum in the Philippines. So by the way, I'm Joseph Valencia, a research student, graduate student from Niigata University, Japan, and a researcher from the Research Institute for Tropical Medicine in the Philippines. So to start with, I'll give you an overview of schistosomiasis in the Philippines. So Schistosoma japonicum still affects 12.4 million Filipinos in 12 regions of the country. So it is still an important public health disease and is endemic in 28 provinces. And there's an estimate of 2.7 million which are directly exposed to contaminated waters. And our national survey as of 2014-2018 focal surveys using CatoCats is 4.8%. So as you can see in the photos in the map, with red colors, so this is a distribution of schistosomiasis all over the Philippines, with the intensity of red color is directly proportional to the intensity of the prevalence. So as you can see, like the focal areas of this infection are mainly here in the middle of the Philippines, which is like Leyte. And this is where our study site is. So current approaches in the Philippines includes preventive chemotherapy, innovative intensified disease management, veterinary public health services, provision of safe water and sanitation and hygiene, which is WASH, and vector ecology management. However, in part of the vector ecology management, we're still, as what like Sandra presented, in the Philippines, we're still like reliant as well in the looking into like intermediate host snail and then examination under microscope, which is like very labor intensive. So to give you the context of how this transmission of schistosomiasis takes place in the Philippines, so this is the areas where schistosomiasis is endemic. So people are doing their daily chores in the river, washing clothes, bathing, and these areas are mostly agricultural. So people are planting rice in these areas. And little did they know that the areas that they're like working with has the presence of the snail, which some of it were infected with schistosoma, and it like in the end they will be like infected, but they don't have a choice because their environment is where they live. So they need to like go to the rice field, plant the rice, or else they cannot eat. So it's really like the disease of the people who are like working in close with these environments. So as you can see in the left most of my side, this the life cycle of Schistosoma japonicum. So here, water buffaloes and dogs are included because Schistosoma japonicum is zoonotic. It's one of the reason why control for this parasite is quite difficult, because other than like humans, you need also to consider like the animal sector for this one. So water buffaloes and dogs are just like among the other host for this parasite other than humans. So infection takes place in these areas through the cercaria that were released by the snail. So the snail is the first intermediate host where they get infected from the egg that were laid down in these areas. So identification of areas where this parasite can be found would give these people would help these people because we can do like environmental modification, building bridges so they will not like wade in these waters or clear these areas to like get rid of these snails. So yeah, so what is environmental DNA? This were already explained by Sandra in her presentation, so I will not explain more of it. But I would like to emphasize that in this study, we use the eDNA to detect the intermediate host snail of Schistosoma japonicum in the Philippines, which is Oncomelania hupensis quadrasi. and as well as Schistosoma japonicum from soil samples. So usually we just like we use like water samples, but in this study we use soil samples. So this approach supports sensitive and invasive identification of microhabitats of the snails and the transmission risk areas. So the question is, why soil? So to give you an ecological rationale for the using the soil, I'll show you the life of the snail. So Oncomelania hupensis Grossi is amphibious, so it stays in water and spend some of its time as well in moist soil. So it prefers wet terrestrial microhabitats like riverbanks, rice field margins, and exhibits limited movement and clump distribution. So making soil sampling effective for localized detection. So if that area, if you know that area is already like having that snail, if you come back like the next year to collect again, it's still on that area. So there's a limited movement and clump distribution. So snail site will stay most of the time as a snail site. So infected snails can contribute to environmental presence of Schistosoma japonicum DNA in these soil-associated habitats. And soil enhances as well eDNA persistence because there are some studies that shows that soil better preserved eDNA. So if you look at the photo on the rightmost of my slide, you can see the development of Oncomelania quadrasi. These were observed in the laboratory. This is already a second generation bred Schistosoma japonicum. So we observed that from like early stages, they are highly aquatic, and after like 6 weeks up to like 6 months, they are highly amphibious. So there's a high probability that these snails will move into the soil near the water areas where these snails can be found. So that's the rationale why we use like soil samples. So we did our study in Ikiran. Ikiran village in Leyte, Philippines is one of the endemic village in the Philippines, endemic for Schistosoma japonicum. So before we did a human prevalence survey, and we found that 25.2% of the people living in the village were infected with Schistosoma using triplicate catocats. So showing you in the rightmost of the village in the map on the in the upper area. This is like an irrigation canal connected to a natural bodies of water in the rightmost, which is like a natural stream. So we did a cross-sectional study conducted in this village in two sampling phase. So we did like phase one in December 2023. So the main aim of phase one is to standardize the collection. So because we don't know how far the reach of eDNA, how far from these water bodies could we collect like samples. And then phase two, after standardization, the collection, we will rule out in all these collection sites, the 30 sites. So here, as you can see, like we did in phase one, phase one. So here we did like an outward collection and with 0.5 meter interval because we want to know how far we can detect like eDNA. So here, based on like phase one, there's no significant difference here in the distance and in the distance from water course. So we decided in phase two to focus the collection here, just here in within 0.5 meters along the water course. So here we did collection of 100 gram of soil per point with standardized handling and cold chain transport. So then eDNA were extracted using alkaline, lysis, ethanol precipitation, and commercial soil DNA kits. And then we did like multiplex detection of Oncomelania grossi, which is the Soma japonicum via mitochondrial CO1 gene. This protocol were already optimized by two previous studies and using qPCR and digital PCR. We also did like parallel molecological survey and morphological identification of the snails. And we did also measurement of soil parameters, pH, moisture, temperature. And then we analyzed the differences within this um, parameters using statistical tests, percent agreement, and we also did GIS mapping. So for the result, so in phase one, we found that, as I said before, we found that we detected successfully the eDNA in soil within 2.5 meters from the water course with no significant difference between eDNA positivity and distance. So we assume that within 2.5 meters, the detection of eDNA is the presence of eDNA is homogeneous. And here you can see that eDNA were detected in 3 out of 6 sites in phase one, because in phase one we only did in 6 sites. And then for Schistosoma japonicum, surprisingly, we detected in 4 out of 6 sites, because previous study only did this detection in snails, but now we included the Schistosoma japonicum, and we detected as well Schistosoma japonicum in soils. So comparing to malacological survey, which only detected 3 out of 6 for the snail, and 1 out of 6 sites for Schistosoma japonicum. So here's the table. And in phase two, we tested all the 30 sites, and we found positivity of 20%. That's 6 out of 30 sites for Schistosoma Japan for Oncomelania for the snail. And 3 out of 30 sites is 10% for Schistosoma japonicum. So all sites with Oncomelania hupensis quadrasi by malacological survey were detected by soil-based eDNA method. And Oncomelania hupensis quadrasi and Schistosoma japonicum eDNA were detected in 3 out of 6, which is 50%, and 3/3, which is 100% respectively. So if you look at the map, so we map here, the red represents the presence of the snail. So the leftmost is malacological survey comparing with eDNA system on the rightmost. So the red is the areas where we found the snails in phase two by malacological survey, and no sites were found to have like Schistosoma japonicum in the snails. Whereas if we use eDNA, we found more sites where we found more sites of Oncomelania hupensis quadrasi, the snails, and also sites where we found Schistosoma japonicum. Yeah, however, as Sandra's as earlier presenter said, that this system can not also differentiate from egg Cercaria. So, but as long as there's a presence of snail and presence of the parasite, there's a high probability the transmission happened, because whether it's from egg or whether from Cercaria, with the presence of the snail, these areas could be a transmission site. And interestingly, the areas where we found a lot of snails eDNA and parasite eDNA are in the area where there's a natural stream. So there's a supply of water whole year round, and unlike this irrigation area on the uppermost part, these are like controlled areas where they can stop the flow of the water. So as you can see, these areas are really like ideal to be like snail habitats. And for the edaphic factors, pH, moisture, and temperature, we found that only pH have significant difference among eDNA positive positive areas and negative areas. So soil pH in the sites examined ranges from 4.5 to 7.4. So to summarize my study, so it shows that soil-based eDNA detected Oncomelania Grossi and Schistosoma japonicum across multiple distances from water courses in the study area. So positive detection corresponded to potential transmission and microhabitats, including sites where organisms were not directly observed. So eDNA results showed that moderate to high concordance with malacological survey, supporting complementary use in surveillance. So detection was successful up to 2.5 meters from water banks, indicating spatial persistence in adjacent terrestrial, aquatic, and transition zones. So persistence may be influenced by soil properties that stabilize DNA, including moisture, organic matter, and fine particles. So in phase 2, it showed the broader scale applicability with high agreement, 90 up to 100%, despite the reduced positivity due to environmental heterogeneity and dilution effect. So overall, soil-based eDNA demonstrate a higher sensitivity than traditional surveys. So particularly in low density and cryptic population, improving detection of hidden transmission foci. So I think that's all. And before I end, I would like to acknowledge the following. Thank you very much.
A huge thank you to you, Joseph. Fascinating work and a fascinating presentation, and really giving opportunities and a practical way to detect transmission where maybe water sampling alone might miss the picture. So thank you so much. We've learned a lot. More questions coming in, of course. Before we delve into those, I'll hand over to you, Mark. We're going to hear from you a lot more about water-based eDNA approaches, and perhaps answering some of those very practical questions that have come in through the chat. Over to you, Mark. Thank you.
Thank you. So I'll be sharing my screen. Is it okay?
Perfect.
Okay.
Thank you.
Thank you. So good evening, everyone from Japan, and on behalf of our team from the Niigata University, I'll be presenting our work on water-based environmental DNA surveillance for spatiotemporal assessment of schistosomiasis, an innovative approach for public health action in the Philippines. So I'd like to thank also Global Schistosomiasis Alliance and International Society for NTDs for this webinar. So the cases of schistosomiasis in the Philippines was already discussed by my colleague Joseph. And so I'll be focusing more on the application of environmental DNA surveillance, specifically the water-based. So as you can see here in the life cycle of Schistosoma japonicum, most of its life cycle stages occurs and is dependent on water or aquatic environments, especially because of the snail intermediate host, which is, as mentioned, is amphibious, which has a life in land and in water. So the current practice of snail monitoring in the Philippines is done by actual observation of snails in the endemic areas. However, the workforce for this kind of job is declining as less attention is being given for environmental surveillance. And also, the presence of Oncomelania hupensis quadrasi in the environment is very difficult to identify, especially for the untrained eye. And as you can see in the picture, is not easily detectable because it blends well with its environment. And also, the size of the snail intermediate host, the Oncomelania hupensis quadrasi, is around 3 to 5 millimeters only. And for the untrained eye, this size and this morphology is especially hard to be distinguished among other snail present in the environment, especially of Limnaea, Jogora, and Sabulina species. So on the right side of your screen, you can see the distinguishable characteristic of the snails. So, One Health approach offers a holistic framework, and it emphasizes the importance of environmental and zoologic factors, not only the human factor of the disease, to effectively manage and potentially eradicate it in the future. And this is where environmental DNA comes into play. The basics of environmental DNA was already discussed by my co-presenters. And so in 2019, a pioneering study that was done by our colleagues from here in Japan and also from the University of the Philippines, Diliman, have established the protocol for the use of environmental DNA technology for detecting both Schistosoma japonicum and its snail intermediate host OHQ. So we built on this study, and we further develop this technique so that it could be more field applicable, or at least semi point of care. So we refined and validated, and we applied a multiplex qPCR assay to simultaneously detect the snail intermediate host and the parasite. And also we use those data to assess the temporal and spatial patterns of the parasite in a communal water sources. And we also generated a hazard map for the potential transmission and contamination sites based on eDNA data. This is to support a evidence-based control strategies for schistosomiasis in the Philippines. Excuse me. So we conducted the study in Ekiran Village in Alang-Alang, and we selected 30 sampling sites based on geographical features and access to water. So in selecting the points, we decided to select the points that are adjacent around 20 to 100 meters in distance. And we also conducted the sampling from July in 2023 up to March 2024 in a periodic manner. So for sample collection, we collected 3 samples of 500 milliliters each site. And then we also collected GPS data and water parameters such as pH, temperature, conductivity, and total dissolved solids to correlate the ambient conditions of the water where the snail intermediate host and the parasite could potentially be inhabiting. And also to compare environmental DNA performance with the conventional methods that we are using for surveillance. So we use a time sampling method. So for the conventional method, we employed snail examination methods where we crush the snails so that we can see the infection in snails in each sampling point. So we note that those places where we found infected snails, if they have the presence of sporosis and cercaria under the microscope. So for environmental DNA sample processing, we employed the gravity filtration system by Oka et al. And we use Sterivex filtration method by Wong et al. So, for field applicability, we employed a mobile probe-based real-time PCR assay. So the thermal cycler, the machine that we are using to analyze our sample, is very compact and small, around this size, and we can perform the analysis benchtop. Just connect it to a tablet or a small computer. So we specifically targeted the cytochrome C oxidase 1, which is a mitochondrial gene for both OHQ and SJ. Because according to studies, the structure of mitochondrial DNA, given that it is in ring form, the structure is more robust against degrading components in the environment. As we expect that the water samples that we will gather is dirty. So we compared the performance of environmental DNA system and the classical methods, and as you can see, we see significant agreement with 71% for OHQ and 85% for SJ, with statistical significant difference. Noting that qPCR is more, or the environmental DNA detection system is more sensitive, given that it has detected more positivities compared to the conventional methods. So as mentioned, we also tested the water quality in which we found out that pH and temperature is particularly significant for the detection of SJ positivity. And we can correlate this with the life cycle of the parasite, given that based on previous studies that the parasite and the snail intermediate host requires a an ambient condition for it to be persistent in the environment. And additionally, this is also based on our experience in the lab when we maintain the life cycle of the parasite and the snail intermediate host, that they are quite sensitive to sudden changes in the ambient conditions. So we also descriptively associated the detection with ambient conditions such as rainfall and temperature. And as you can see, during the months with lower temperature and higher rainfall is when we were able to detect more positivity for both the parasite and the snail intermediate host. And again, this correlates to the uh, specific requirements of both the parasite and the snail intermediate host to thrive in the environment. So for the implications of eDNA detection, so this is what we use to infer our results. So most importantly is the areas where we can find both the snail intermediate host and the parasite. So this is where we consider it as an active transmission area, and of course the safe areas where we can use eDNA data to inform the people living in endemic areas that these places are safe for communal use, and you can use these sources without worry of being infected with schisto. And it is also important to note the potential transmission area and the contamination areas, as introduction of the parasite or contamination in potential transmission areas where we can find the snail is uh, probable to be an active transmission areas, and also for contamination areas where people how do you say this, continually contaminate the area. If flooding happens or the transfer of the snail in those frequently contaminated areas, active transmission could also be possible in those areas. So this is our map based on environmental DNA during the quarters that we did the environmental sampling. And as mentioned, more positivity were detected in during December and March, where an increase of human activity, given that these times are, if I remember it correctly, harvest season. So human activity, most specifically farming, is more prominent. And so environmental contamination could happen. And based on the collective hazard map from the previews, as you can see, the places where we able to detect the snail intermediate host and the infected snail intermediate host, we were also have been able to detect using environmental DNA, and most especially, as you can see here in site 1. And then most of the distribution of the snail intermediate host and the parasite happens along the natural stream, as you can see on your right. And the fluctuations in the distribution of OHQ and SJ likely corresponds to life stages of them, as mentioned before. So in summary, fluctuations underscore the influence of meteorological variables and water quality on the potential spread of Oncomelania hupensis quadrasi, and by extension, the parasite as well. Environmental DNA detection system demonstrated sensitivity compared to the conventional method, wherein we were able to detect more positivity in adjacent areas. And also, we were able to complete the activity in around 4 to 5 days, with around 4 people working during the field work. And we were able to assess 30 sites in those very limited time. Whereas conventional observation methods, by experience also, is more time consuming and it requires greater number of human resources. So for the environmental DNA detection system complements the Sustainable Development Goals target, particularly goal number 3, 3.3, which aims to end neglected tropical diseases, and 3D, which aims to strengthen the capacity of all countries. And furthermore, this also complements target number 6, which is the water and sanitation, emphasizing the interconnectedness of water quality, sanitation, infrastructure, and parasitic disease transmission. So for our future plans, as mentioned by the first presenter, environmental DNA-derived materials can also be utilized in a range of downstream metagenomic analysis, such as we can pinpoint who among the around 40 species mammalian reservoir hosts is mostly contributing to the environmental contamination. And also, eDNA-based monitoring could inform mathematical models to facilitate the development of a more robust hazard maps to inform control strategies, not only in the Philippines, but also not only for the Philippines, but also other countries, and not only for schistosomiasis, but also other neglected tropical diseases. So, we are currently expanding the scope of our study, and hopefully in the future we can submit this for a policy brief, so we can apply this this innovative technology to be used for environmental surveillance in the Philippines. And as mentioned again, we can also use this for other pathogens that are actively transmitting in the environment. So before I end, I'd like to acknowledge our team from the Research Institute for Tropical Medicine, the local government unit of Ekiran Alang-Alang Leyte, where we conducted the study, and also our collaborators from Australia, National University, and QIMR. Should you have further questions, and if you would like to see more about our results, you can scan our published studies here, and you can communicate with us through email. Thank you.
Thank you so much, so much, Mark, and big congratulations to you, to the team, to Professor Marcello Sato as well, who couldn't be on the call today, but very much spearheading this work. It's been a fascinating session, a fascinating hour, and you've triggered a lot of questions. Perhaps we could start off by just looking a little bit more at some of the questions that have come through about the standard procedures, or operationalizing a little bit more of I know you've touched on it, but some of the methods and details of those procedures. So I've sort of ranked them according to different topics of what you, a huge amount of work that you shared with us. But before we do that, Bruno Sangor saying thanks for this research. Jérôme Boissier, thank you so much for these brilliant presentation. And Roberta Caldera saying congratulations, Mark and Joseph, great work. Congratulations, Marcello. And of course, very jealous of Patricia and yourselves, who are going for lunch tomorrow, and we wish we could join you. So enjoy. Well, we've got about half an hour for questions, and the first one that sort of springs to mind and came from a number of colleagues. Adam Czaplinski, Bonnie Webster, and Tom Pennance were really asking about the filtration methods that were used in the absence of a real consensus in the literature. Could you say more about the filtration method? How much water needs to be processed? Any clogging that might happen, and also where in the water body would be the best water sample to be taken from, which kind of pump you're using. So basically as much detail as possible. And if there are things that we need to look out, that need to be looked out for according to different environments, perhaps, or some gray areas in this topic. I'm not sure who wants to go first, so over to the three of you.
Thank you for the question. Talking about Brazil, we use it 500 milliliters per site in triplicate. This usually we use Nalgene filters. It's like a filter to filter like medium from the cell culture. And usually we had 1 or 2 membranes per site, but there are some places that we had a lot of membranes, so more than 10 or even more. And in these cases, we decided to do the DNA extraction from 2 membranes by chance. But we know that this is not the ideal scenario. So maybe in some areas we need to explore another ways to filter the samples, because we had a lot of clogged in the filter and a lot of membranes. Basically, is this.
Brilliant, thank you. Joseph and Mark, would you like to add to this?
Sure. So for our method, so we are using Sterivex, and at first, we are experiencing a lot of clogging using that filter. So we went on standardizing our water sample collection. So as well, luckily for us, most of our sampling sites have clean water. So because Oncomelania hupensis quadrasi is very sensitive to how do you say, very polluted habitats. So yeah, but it's still possible to have clogging, and to work around that. Say for example, the filters is very clogged and cannot filter anymore. So we take note of the how much water volume have we filtered, and we included in the further analysis. So that are those are the workarounds that we do for water filtration.
Brilliant, thank you. There's been a follow-up question by Marjolin Torians, and it has given me an idea, which it was already very difficult. And thank you so much, particularly given the time zones, to bring everyone together here. But maybe there's scope for like a more informal, very brief meeting to really dive into the details of these operational elements. Marjolin was just saying, when you say you use multiple membranes, does that mean that when after X amount of your half a liter sample clogging happens, you switch over to a new filter to continue filtering the same sample? And then after DNA extraction, merge these 2+ samples and consider them as one sample?
Yes, is this, but when we merged them, we had a huge volume of eDNA, so it loses sensitivity, maybe. So
got it.
that's
Sorry.
the problem. So we had a lot of DNA extractions, so a large volume of eDNA, and maybe it can be used in a large volume in the qPCR.
Got it. I'm just going to pause that topic there for a moment, because we also had some really interesting questions about something you mentioned earlier on in your talks, and that was all around the LS-PCR. Na Ajale Frempong, also Marjolaine Torians was asking, what exactly is LS-PCR? Was it useful for the detection of schisto-eDNA in the field from water? And also, how might it compare to qPCR LAMP, LS-PCR, or a regular PCR? What might be best in your experience for field samples?
The LS-PCR is a normally PCR, like the reagents and all of this, but we use it at different, a low stringent temperature during the cycles of the PCR. So it's used in the National Reference Service with the snail samples to distinguish between the non-infected and infected snails. And it's very cheap because it's a basic PCR. So we tested this, and it works with lab conditions, lab samples. But when we did it with the field samples, we did not have the same sensitivity. Maybe because the technique is less sensitive, or they have different targets from the qPCR and the LAMP assays. But we had the similar results from qPCR and for LAMP when visualized by electrophoresis. They are very comparable, but we did it with a small number of samples, field samples. It's not a huge number, but we decided to do in this new ongoing working just with qPCR.
Brilliant. Joseph and Mark, would you like to add anything to this?
Yeah, based on my experience. So I also compare like two methods. So I compare digital PCR and the regular qPCR. However, my detection with qPCR is quite higher compared to digital PCR. So I think one of the things that we're looking into is the use of reagent, because most of the digital PCR have like a very specific reagents where we cannot like change. Unlike in qPCR, we use a specific pack for environmental samples, because we know that environmental samples have a lot of contamination, a lot of like inhibitors, especially from the we collect from the environment, so which may hinder the reaction to happen. So yeah, based on our experience, the use of that specific environmental pack and qPCR works well compared with digital PCR.
Great. Thank you so much. I'm just going to move to a question particularly, it says here for Mark, from Tom Pennance. Any thought on whether the increased detection of Schistosoma japonicum eDNA at lower rainfall due to the smaller size of water bodies or lower water level or decreased flow rate? Was it due to that? Or was less water disturbance from the rainfall changing the distribution of eDNA in the water? And should eDNA sampling protocols be carefully scaled to water body sizes and seasons to improve detection across the year or sites?
Yes. Yeah, we also found that there is how do you say this? There is a significant effect on the size of the water body, because as water rises, um the environmental DNA is getting more diluted in the water bodies. And so that's why we did the temporal assessment. But based on this, based on our results, most of the positivity is during the rainy season. So the water level is a bit more higher. But also, activities during the time also increased. And so those potential contamination from maybe mammalian host, or it's the active shedding of cercaria from the snail, could have potentially increased the detection in those areas.
Joseph, over to you.
Yeah, I would like to add, and Mark. Actually, it's a very interesting question. So whether like shrinking water, how does like rainfall affect the like eDNA detection? But also interestingly, in the biology of the snails, it's also related in like cycles of rainfall and dry season. So during like high rainfall, those are also the time that they would like to breed. So yeah, maybe these activities also influence the presence of eDNA in the environment. So um breeding and also other activities of the snail that are like related with the season.
Brilliant, thank you. On the theme of detection and life cycle, a few questions here that came up actually during the soil-based eDNA approach presentation, but probably apply to all of them. Najele Frempong was asking, did you find snails that did not shed cercaria over several days, and what could account for this observation, or what impact might that have? Bonnie was also asking, how long does eDNA from snails or cercaria persist in the environment? Connected as well to Bruno Senghor's question, what was the limit of detection of DNA according to the distance of water courses for the soil-based detection? So just generally, what needs to be considered, or what impact of the cercaria in the environment?
So a lot of questions. Which one first? Oh, maybe I'll answer the technical one, the distance. So actually, from the 0.5 meters up to 2.5 meters, there's no significant difference. So for the detection limit, I think I base on CQs. So CQs are also like well distributed. There's also no significant difference. I also expect that the one near the water courses will have like higher eDNA, so maybe the city value would be like lower, but uh the city value are almost like distributed from the 0.5 meters up to 2.5 meters. So we assume that all these areas, so they have like homogeneous in homogeneous eDNA in this area from 0.5 to 2.5 meters from water courses. So I think I remember the snail. I think yeah, for in the case of Oncomelania hupensis Grossi, so it's just a tiny snail. So what we're doing in the lab is we crush the snails individually and examine it under microscope. So if we found like even like the presence of like the young stage of infection, we can still identify that if there's some like cercaria that can be found. So we're not dependent on shedding. So we understand that sometimes shedding could like miss positive snails, especially if it's like in early stage of infection. But for us, like we crush the snail and examine it microscopically. So I think the things that we like say it's positive, it's almost like 100% like positive.
Brilliant, thank you. Any further comments, Sandra and Mark? There's a really interesting addition in the chat. For those who can't see the chat very well, Roberta Caldera was adding that in answer to a previous comment, we don't interpret these results as false negatives, as schistosoma DNA found maybe from Miracidia due to fecal contamination or cercaria. In these cases, if no mollusks are found at the collection site, but schistosoma DNA was found, it may be due to fecal contamination, which is why the team is performing qPCR to detect the presence of human feces in the same water collection points where schistosoma was found without mollusks. Sorry, I'm just sometimes repeating the comments for those who can't always see the chat on their phones, etc. Thank you so much for that. And I guess this was formulated in a far more eloquent question than mine by Jérôme Boissier, just to say mollusk population and schistotransmission can be highly dynamic. Is there not a risk that eDNA methods obscure these dynamics due to eDNA persistence in the environment? And how long does eDNA from snail or cercaria persist in the environment?
I'm not sure I have the answer for this question. Maybe I think it's very according to the water conditions, the temperature, and things like this. But yeah, we are not sure yet about how long and how the distance between the positive site and the DNA is found.
Brilliant. Well, oh, sorry, Mark, after you.
I'd like to add on that. So we found several studies that environmental DNA is persistent for around maybe around 1 week, but still it is dependent on the water conditions because more pollutants could potentially damage the DNA faster. However, if there is a direct presence of for example, the snails, so every day they could shed environmental DNA, and so we can be able to detect their DNA even if having a lot of contaminants. Um, but for again for the ideal conditions, let's say for example the water body is less contaminated, environmental DNA could persist for around a week, given that it's not directly how do you say this, directly exposed to sunlight, because we know that UV can destroy DNA as well. And for the case of Oncomelania hupensis quadrasi, its habitat is characterized mostly by lush vegetation, so it's covered from sunlight, and also perpetual wetness. And also, as I mentioned in my presentation, the persistence of Oncomelania hupensis quadrasi in environment is dependent on the water condition, as they are sensitive to contaminants in the environments. Thank you.
Brilliant. So we've heard a lot about the technical parameters and questions that needed elucidating. We do have about 10 minutes left, and this webinar originally promised transforming schistosomiasis control with eDNA, so not just One Health innovations, but also public health implications. So maybe in the last few minutes, we could broaden the lens a little bit and turn to that from your experience. So what, over the last decade or so, technological innovation and approaches, as you said, Mark, not just for schisto, but across a wide range of diseases. We really saw an uptake of eDNA in the COVID pandemic, also for things such as norovirus, etc. And where do you see immediate and really aligned impact for an eDNA approach, whether that be regionally or, but at the same time, where are the bottlenecks that exist from a public health perspective, and what would you like to see more of to see this approach applied a lot more in a public health program or approach?
I think it can be used for initial screening, maybe pointing some areas where the snail surveys may be done, and also the stool examination in people. So, like more an initial screening for appointing these areas, because as I said in my presentation, here in Brazil, we had a lot of areas with low indemnity and low parasite burden. So it's even more difficult to found a positive snails in this context. So maybe with the eDNA, we can appoint for principal areas, settings that can be more explored for public health. And then I think it's it.
Yeah, I think eDNA would be like very beneficial in public health because like we're looking yeah, eDNA looks into like the commonality, like community level, like the bigger area where the shared areas where this community is living. Um, yeah, other than Schistosoma, actually in the Philippines, some study actually uses like wastewater to see like antibiotic resistant bacteria. So yeah, it broadens the like surveillance part of public health. And we could also use it actually to like do surveillance for like emerging or emerging diseases. Yeah. I think that would be like future of eDNA. Also like maybe as a tool sensitive to a sensitive tool to know the transmission interruption elimination, because we know that going to elimination it takes a lot of work, but the real work is after elimination, how we ensure that the disease is no longer existing in the area, especially those environmental disease that are linked to the environment, just like schistosoma. So I think eDNA is really a big and sensitive tool to do to apply in those areas. Yeah.
Yeah. Um, for the sustainability impact of environmental DNA detection system, as Joseph mentioned, we can apply environmental DNA technology in nearing elimination or places with low endemicity. And also we can incorporate environmental DNA data to spatial or geospatial modeling, so we can predict the spread of the disease more accurately with the use of eDNA technology. And so it could affect our strategies into eliminating the disease, not only schisto but others. Thank you.
Fantastic. And just coming back to specifically schisto and tying in with a question from David Rollinson asking, do you feel that eDNA approaches could now be recommended in studies to confirm interruption of transmission? Do you just bringing together everything we've heard over the last hour and a half, do you think that we're there? Do you think that the approach could benefit with a few more implementation pilot projects, for example? Where along the spectrum of full integration into the elimination agenda is this approach for Schisto?
Yeah, but I think we need more evidence on this because I'm thinking like we need to know like whether eDNA is like directly linked to the reduced cases in humans and animals. So yeah, I think we need to conduct more studies, but as of now, the evidence I think is leaning towards like it's a promising tool. But for me, there's still a need for more like bigger evidence for us to really like establish and standardize everything.
Brilliant. Oh, Sandra, after you.
No, I just agree with Joseph, and I think we are working on this, but we still have to do more field working to set this as a public health tool. But it's promising.
Fantastic. So moving away from the technical scientific aspect to more real world applications. And clearly there was a hugely engaged audience, just anecdotally around this webinar, lots of interest, a really dedicated community of practice in a way that really wants to learn and collaborate. Kennedy Kama sort of articulates exactly my thoughts. I suggest that these studies be extended to the African settings. And so if there are any collaborations coming together, thanks to the discussions today, it would be really lovely to be in touch and keep in touch. Lots of attendees putting their email address in the chat, and everyone completely agreeing that as Hala Al-Bashir says, thank you for the discussion. It opens a lot of doors. Dio Gracia saying very informative presentations. Absolutely. There's certainly feels like there's a lot of momentum behind this approach and this budding field. If you have any final words that you'd like to share before we bring this webinar to an end, maybe any calls for partnership, more funding is always nice. Just anything you'd like to see more of in your work for your highly collaborative teams, please, over to you for the final words of the session.
I'd just like to thank you the opportunity to present our research here from Brazil, to represent my research group. And we are also always open to collaborations, so please feel free to contact us. And thank you very much.
Yeah. Thank you. Thank you for this opportunity also to present our work. And in advance, I would like to say thank you to the funders because we aspire to work more in this and maybe apply this in a wider scale and gather more evidence regarding eDNA and ways how we can apply this in the future that could benefit the public health. So again, thank you very much. And for anyone who would like to collaborate, please contact us or our professors in the email given in the presentation. So thank you very much again.
Fantastic.
So, again, I'd like to thank on behalf of our team, GSA and ISNTD for the opportunity to present our work. You have given us the avenue to share how environmental DNA could transform One Health approaches to disease elimination. And yeah, we would be glad to collaborate more, and not only for the Philippines but also other countries with schistosomiasis. And I'd like just to comment on the comment that was shared that eDNA technology should be done also in Africa. So our professor Marcelo Sensei already applied environmental DNA. I think they published their results in 2017. So they did a study in Madagascar targeting Mansonite. Thank you.
Fantastic. Wonderful. And just also to highlight that the Global Schistosomiasis Alliance has a lot of very dynamic working groups. So you can also connect as part of those. Adam Chaplinsky says, thank you very much for fantastic presentations, very informative and lots to take in as this team works on the eDNA project right here in London. So a method applied everywhere. And because we love eDNA at ISNTD, if you want to hear more, please join us. In fact, this Thursday, April 23rd, we have our annual vector-borne diseases event, ISNTD Bites, and another wonderful application of eDNA. We'll be hearing all about that for integrated vector surveillance and detection of invasive species, mosquitoes in this case. Other than that, I hope that we get to cross paths very soon again. Thank you so much for giving us your time, both very early and very late. And a huge thank you to the incredibly engaged audience for all your wonderful questions. I'm sorry that we missed a few. There were still a few outstanding, but our speakers have said loud and clear, don't hesitate to get in touch. On that note, I wish everyone all the best. Thank you for your time, and let's speak very soon. We'll be really excited to see where your work and your projects go next. Thank you, everyone. Take care.
Thank you.